As a pioneer in immunotherapy, Transgene is using its InVir.io platform to develop innovative treatments that can attack cancer cells directly, oncolytic viruses. InVir.io is based on a patented and highly attenuated strain of vaccinia or cowpox virus. Its genome has been modified to replicate only in cancer cells. The anti-tumor efficacy of these oncolytic viruses is based on combining three complementary mechanisms of action. When administered to the patient, these viruses infect tumor cells where they selectively replicate. Viral multiplication then results in the targeted destruction of the tumor cells that have been infected. This is called oncolysis. In addition, our oncolytic viruses have been engineered to carry a full therapeutic arsenal of anti-cancer weapons, such as antibodies, cytokines, or enzymes. As these therapies are produced directly in the tumor, their efficacy is optimized and healthy cells are preserved. As a result of these activities by our oncolytic viruses, the tumor once again becomes visible, reactivating the patient's immune defenses, which then are focused on attacking it. With these three complementary mechanisms of action, Transgene's InVir.io-based oncolytic viruses represent a true innovation in the fight against cancer and in preserving the patient's quality of life. TG6050 is a new oncolytic virus from the InVir.io platform. Two genes coding for therapeutics have been incorporated into its DNA, namely interleukin-12 or IL-12, a cytokine that triggers a powerful anti-tumor immune response, and an anti-CTLA-4 antibody. Administered intravenously, TG6050 selectively replicates within tumor cells. This wakes up the immune system and leads to the production of the two therapeutics. Interleukin-12 and anti-CTLA-4 antibody produced by cancer cells also promote the induction of the immune response. IL-12 multiplies, activates, and transforms naive lymphocytes into lymphocytes capable of attacking the tumor, and activates NK cells, which, in turn, destroy tumor cells. In parallel, the anti-CTLA-4 antibody restores the immune defenses within the tumor by liberating so-called costimulatory molecules and by neutralizing the regulatory T cells that inhibit the immune system. As a result, TG6050 is able to trigger an immune cascade that allows the body to fight against the tumor and metastases. This novel, highly targeted immunotherapy approach could represent a major advance in the treatment of many solid cancers, including lung cancer, by avoiding the side effects associated with systemic administration of IL-12. With TG6050, Transgene is pioneering a much-needed new approach in the fight against cancer.